Hello, dear students. In this part of the unit, we will study the major biological molecules found in living organisms. By the end of this lesson, you should be able to categorize biological molecules based on their structure and function, identify the components of enzymes, and explain the roles of these molecules in metabolic processes. Keep these learning targets in mind as we move through the next set of slides. The main question we aim to answer is this: how do the structures of carbohydrates, lipids, proteins, and nucleic acids determine their functions in living organisms? As we go through each biomolecule, pay attention to how its shape, size, and chemical properties allow it to perform specific roles in the cell. A biomolecule, also called a biological molecule, is any molecule found in living organisms. The four major classes of biomolecules are carbohydrates, lipids, proteins, and nucleic acids. Most biomolecules are organic compounds made primarily of carbon, hydrogen, oxygen, and nitrogen. These four elements alone make up about 96% of the human body. Despite being built from similar elements, biomolecules differ greatly in structure, and these structural differences lead to very different functions. Carbon is a versatile component of life's molecules because of its unique bonding abilities. A carbon atom has 4 valence electrons, allowing it to form 4 strong covalent bonds with many different elements, including hydrogen, oxygen, nitrogen, and other carbon atoms. This makes carbon an ideal backbone for complex biomolecules. This slide presents an overview of the 4 major biological macromolecules. You can see how carbohydrates, lipids, proteins, and nucleic acids differ in size, complexity, and role. Carbohydrates are mainly used for energy, lipids for long-term energy storage in membranes, proteins for structure and function, and nucleic acids for information storage. Carbohydrates are a large group of organic compounds produced mainly by plants through photosynthesis. Common examples include glucose, fructose, sucrose, starch, and cellulose. Many carbohydrates taste sweet, and these sweet carbohydrates are called sugars. The most common household sugar is sucrose. Carbohydrates play a major role in providing energy to living organisms. This diagram shows the basic structure of carbohydrates. Monosaccharides. Notice that carbohydrates often form ring-shaped molecules like glucose. These ring structures allow carbohydrates to easily participate in chemical reactions that release energy. This slide shows 3 important polysaccharides, all made of glucose monomers. Starch is stored in plant cells for energy. Glycogen is stored in animal muscle and liver cells. Cellulose forms strong fibers in plant cell walls. Although all 3 are made of glucose, their different arrangements give them very different functions. Cellulose and chitin are structural polysaccharides that provide strength and support rather than energy. Found in plant cell walls, cellulose have hydrogen bonds which allow it to form strong, rigid fibers. Because humans lack enzymes that break these bonds, cellulose is indigestible and instead functions as dietary fiber, supporting digestive health. Chitin is similar in structure to cellulose, but occurs in animal exoskeletons, such as those of insects and crustaceans, and in fungal cell walls. It is made of nitrogen-containing modified glucose units, which make chitin stronger and more durable, providing protection and structural integrity. Carbohydrates serve as the primary source of energy for cells. They also provide structural support and aid digestion through dietary fiber. Lipids are a diverse group of biomolecules whose common feature is that they do not dissolve in water. The term lipid comes from the Greek word meaning fat. They include animal fats, oils, phospholipids, and steroids such as cholesterol. These diagrams show different lipid structures. You can see long hydrocarbon chains, which explain why lipids store so much energy. Phospholipids have a hydrophilic head and hydrophobic tails, allowing them to form the bilayer of cell membranes. Steroids have a distinctive 4-ring structure, which is critical for their role as hormones. Phospholipids are major components of cell membranes. They form a bilayer that separates the cell's internal and external environments. This image shows how lipids are arranged in biological membranes. Their structure allows membranes to be flexible, yet stable. Steroids are lipids with ring-shaped structures. They function as hormones and as components of cell membranes. Saturated fats and unsaturated fats are 2 main types of dietary fats that differ in their chemical structure and effects on health. Saturated fats have no double bonds between carbon atoms, which makes them solid at room temperature and commonly found in animal products such as butter, meat, and dairy, while unsaturated fats contain 1 or more double bonds, are usually liquid at room temperature, and are found in foods like vegetable oils, nuts, seeds, and fish. In general, unsaturated fats are considered healthier because they help support heart health, while excessive intake of saturated fats may increase the risk of cardiovascular disease. Lipids store energy, form cell membranes, and provide insulation and protection. They also help absorb fat-soluble vitamins, like vitamins A, D, E, and K. Proteins are the most abundant macromolecules in cells. They are made of amino acids and perform a wide variety of functions. Human diets obtain carbohydrates, lipids, and proteins from many food sources that support energy, growth, and repair. Carbohydrates are found in foods like rice, bread, fruits, vegetables, and grains. Lipids, or fats, come from oils, nuts, seeds, dairy products, avocado, and fatty fish. Proteins are obtained from animal sources such as meat, fish, eggs, and milk, as well as plant sources like beans, lentils, and tofu. Proteins are composed of amino acid chains that fold into specific shapes. This folding determines how each protein functions. Proteins perform several essential functions in the body. Enzymatic proteins, or enzymes, speed up chemical reactions by lowering activation energy, such as amylase breaking down starch and sucrase breaking down sucrose. Storage proteins store important substances for later use, such as ferritin storing iron and casein storing nutrients in milk. Defensive proteins, including antibodies, protect the body by recognizing and neutralizing pathogens like bacteria and viruses. Hormonal proteins act as chemical messengers that regulate body processes. For example, insulin controls blood glucose levels by signaling cells to take in glucose. Receptor proteins, found on cell membranes, bind specific molecules such as hormones or neurotransmitters and trigger cellular responses, allowing cells to communicate and respond to their environment. Transport proteins move substances within the body or across cell membranes, such as hemoglobin, which carries oxygen in the blood, and membrane transport proteins that regulate the movement of ions and nutrients. Structural proteins provide strength and support to cells and tissues, including collagen in connective tissues and keratin in hair, nails, and skin. Contractile proteins, such as actin and myosin, enable movement by allowing muscles to contract and produce body motion. How are proteins made? The central dogma of molecular biology explains the flow of genetic information within a cell, moving from DNA or deoxyribonucleic acid to RNA or ribonucleic acid, and then to protein. In eukaryotic cells, proteins are produced based on genetic instructions encoded in DNA found in the nucleus. Mistakes in protein synthesis can result in non-functional proteins. These errors may cause genetic disorders or cellular malfunction. In sickle cell anemia, a mutation causes the hemoglobin protein to be defective, producing hemoglobin S instead of normal hemoglobin. This abnormal protein makes red blood cells rigid and sickle-shaped, reducing oxygen transport and causing pain, anemia, and circulation problems. Defective proteins can disrupt normal cellular processes. This highlights the importance of accurate protein synthesis. Overall, proteins provide structural support, transport substances, enable movement, and defend against disease. Most importantly, enzymes are proteins that speed up chemical reactions. We will talk more about enzymes later. Nucleic acids store and transmit genetic information. They are composed of nucleotides. These are named so because they come from the nucleus of the cell and are acidic in nature. Each nucleotide consists of a sugar, a phosphate group, and a nitrogenous base. These components form DNA and RNA. These diagrams compare DNA and RNA. DNA is double-stranded, stored in the nucleus, and used for information storage. RNA is single-stranded, active throughout the cell, and used in protein synthesis. The diagrams show the double helix structure of DNA and the complementary base pairing. DNA contains the bases adenine, thymine, cytosine, and guanine. RNA replaces thymine with uracil. In gene expression, DNA serves as the permanent storage of genetic instructions. These instructions are copied into messenger RNA through transcription. The RNA then carries the coded information to the ribosome, where it is translated into a specific sequence of amino acids that forms a protein. Through this process, genetic information is converted into functional molecules, showing how DNA ultimately controls cell structure, cellular functions, and the growth and development of the entire organism. Nucleic acids store genetic information, guide protein synthesis, and allow inheritance and variation. Mutations in DNA introduce variation, which drives evolution. Thermodynamics predicts whether reactions occur, not their speed. Spontaneous chemical reactions can be extremely slow and imperceptible, almost impossible to perceive. Enzymes speed up chemical reactions that would otherwise occur very slowly. Enzymes lower activation energy needed for reactions. They remain unchanged after the reaction occurs. Without enzymes, many life-sustaining reactions would not proceed efficiently. Enzymes are usually named based on their substrate or reaction. Most enzyme names end with the suffix ase. Digestive enzymes are clear examples of enzymatic proteins that help break down food into smaller, absorbable molecules during digestion. For example, amylase in saliva breaks down starch into sugars, Pepsin in the stomach digests proteins into amino acids, and lipase in the small intestine breaks down fats into fatty acids and glycerol. Enzymes are highly specific to their substrates. This specificity ensures accurate chemical reactions in cells. The lock and key model explains enzyme specificity. The substrate fits precisely into the enzyme's active site. Only the correct substrate can bind to a specific enzyme. This selectivity prevents unwanted reactions. This diagram illustrates how enzymes interact with substrates. Notice how the active site holds the substrate during the reaction. Enzymes are sensitive to temperature and pH because these factors affect the shape of the enzyme's active site. Each enzyme works best at an optimum temperature and pH, and conditions that are too high, too low, or too acidic or basic can reduce activity or cause denaturation, stopping the enzyme from functioning. Enzymes are important because they regulate metabolism and maintain homeostasis. Without enzymes, life-sustaining reactions would occur too slowly. As an answer to the essential question, the structure of each biomolecule enables it to perform specific biological roles. Structure and function are closely linked in all living systems. Before ending the lesson, recall the monomers of each biomolecule and their roles. Then, reflect on how enzymes and nutrients support metabolism in living systems. Thank you for listening.